我赖不出去喽。现在不要再讲奇怪的话喽，我线上也都听得到喽。好，因为我要去听隔壁的声音。
对，这不是在秀，那是传统比赛，那个比赛，对，是没有出过的，我觉得我不会尝试。你电脑多？我的环境这么多，然后非常好使，有没有去学？说有机会再说。有啊。还没有多大很大。
那这样你要怎么样？倒掉它吗？
不要，不要吃它。
posted here in the board.
OK。
Both professor.
早上那个需要10号下午再是那个对。先洗澡鸡蛋，然后就其他就是这群收了。
哦，我报名打给我。
啊，
哦好。
哎，今天我们是几点？1点。1点好，那个谁在下面？我们刚刚是那个一起来。Hello。喂。潘神。哦，这一改麻烦，真的在这。哎，我们要一路麻烦你。
自己坐，自己找位置坐。
听起来好可怕。没有，我们今年之后就要把你那个跟带那个合并哦。就说科米出来。没有，可以啦，可以，这样子，给掉你先看。我知道。晚上2点。
我后面可以坐这里，然后我第一段可以坐这里。
勉强可以这样讲哦。OK。我们都一直麻烦他，没有。那我再跟你讲，我们今天晚上去那个，可以吗？
OK， 你就维持在，就是一开始他演讲的时候就维持在这个话题，然后你就可以继续说。
放下来，好不好？
有。OK， 知道。Syuan-Jyun。先拿。这个是到时候他们线上有那个问题，可以看。
对，那你也看见了。这个是礼拜三你有空吗？礼拜三中午。然后我们就会分4个人，那个活动我们分4个人。然后都在中午的那种，就是大概就是12点。对，我们是中午12点，对不对？12点半。
这个是我也可以参与。
你可以啊。
没有教评会。
我们就是今年你们的那个，但是我们以后你就变小评会成员，就这样。
你要说什么？
就来，就帮我们讲。听4个人的哦 ，OK 哦，好了，那可以休息了。
你先帮我打点
这个过程中，我就需要去考1年，先你
帮我跟过。
第一点时间。所以我们是完全是个过渡期，就是把你们
就是投出去吧。
对，这个是当时因为那个这样，OK。
反正有意见我可以书面意见。
可以啊。所以
你看直播没有？直播完它直接就在上面。哦，对。但是你们现在借我过，我要找这个 pen， 还是你调？给你调。我们把他的眼睛放在那个2/3的线。所以它太低。啊？
上次录音声音品质还是有一点。
你说上礼拜4吗？对。上礼拜4我们找到 bug 了，是一只那只麦克风坏了，所以马上当天下午换了一只新的。
好。那个
speaker 都很清楚。
对 ，speaker 都很清楚。我们后来有看直片，就是外面的人很清楚，然后摄影老师也很清楚。
我们这边自己我们的。
好。
可是我们原本这种环境就是最难收音的。
对，
就是
不要用麦克风吗？
不要用麦克风。然后第一个不用麦克风，其实后来我去找厂商跟他讨论，当场讨论。他听什么？
好，我们再给你5分钟。OK， 3分钟不用紧张。好。
这个高。
大家如果还没吃晚餐，haven't finished your stomach, please go ahead.
不想累了，不要听。
OK， 大家认真吃。
对，我知道。
麦克风测试。对。好。麦克风测试，有。OK。待会如果他开始讲话，就把这个后，还记得。
消退式。
这是环境好，
这是这一只，对。
就是我们在同行，我们在线上同行。对啊，大家都听到他们跟他们的这个对。唯一有这样子才会不然就没有。安发是对他需要。
增强它，更多的学生啊，我们也在找去年这里
一个东西，
开始去为40个
地铁另外一个大概都是这样，但是我觉得大概
帮我们知道那个讲或者对策，
不用了，也不用了，我只慢慢讲，我都是在学，因为就是作为一个跨的是所以我们的一起
现在很简单，因为好像都是其实这个直播完就直接放语音，所以什么都不在。
这样可以打到
好像数学系有的专业，还有修改的版，
你现在一个礼拜大概就是13个小时，直播时间。
是在数学系的时候，
现在会第二个讲的有下来讲。
在另外一个学校，对，因为
别的你在衣服不会摩擦到，这是讲口袋。
OK， 好，9位。Okay, I think let's start today's the third job, and then we have the one position open for the new candidate to come over to IBCS, and Dr. Syuan-Jyun will give us a speech, and he is actually our third candidate for the shortlist at the moment, and so now the floor is all yours. It's all 1 hour, and then we will have another 1 hour interview with the faculty members with you, and then after that there will be the half hour you and students together have a certain kind of conversation without faculty members, so you can talk whatever you want, ask any question to the students if you like, and then we have some faculty over here. I just very briefly to introduce you, and then Dr. Lai has visit already, and then Thomas has been here, and then John, you know him?
On Twitter.
On Twitter, okay, you are a Twitter friend, okay. And then another one will be Luo Minghui over there, okay, and Luo Minghui is from the Automobile Science Department as well. And then anyone online at the moment, or do you see anyone? We will record it because this time is our IBCS seminar 3, 4, 5, 6 for the master and PhD students. But some faculty are not IBCS, they are joining with other departments, so they may not have the time here. We will do the recording and then we will share with them for the decision making about the shortlist and the finalize the shortlist. Okay? So let's start and then your turn. So we give the report to
Thank you very much all for coming. It's my great pleasure to be here. So I'm a behavioral and evolutionary ecologist that I'm interested in how animals adapt in a changing world, with a particular focus on the way that the species interact with each other and how they respond to the changing environment. So for centuries, we have always been fascinated by interactions between species. So they are important because of the biodiversity and also the ecosystem functions they provide. Symbiotic organisms in particular that live intimately with each other and can form mutualistic partnership, such as in the clownfish and anemone. But they can also benefit by harming the other, such as the host-parasite interactions. And what's more interesting is that many of these seemingly stable interactions are highly variable in space and time, ranging from mutualistic, commensal, or even parasitic, even within the same pair of interacting species. And so the species interaction really is the foundation that shape the ecology and evolution, and also contributing to our biodiversity on Earth. However, we're in a 6-month species extinctions worldwide, so climate change and habitat fragmentations are two major drivers for the global biodiversity loss. And climate change, of course, is impacting every ecosystem because of the rising temperatures and also extreme weather events. Habitat fragmentations, on the other hand, due to degradations or habitat loss, is crucial because it directly reduces the amount of natural habitats, which links to the rapid human population growth. Well, the insects are amongst the abundant animals on Earth and are commonly found in all habitats and are essential in maintaining our ecosystems. Yet their sharp decline and extinction due to climate change and habitat fragmentations has raised serious concern about ecosystem functions and the fate of millions of species, including we human beings. So to me, I'm obsessed with the insect diversity, but on the other hand, they're also suffering from the environmental changes. So these inspire me to address one single key question. So how does biodiversity persist and function in our human-modified world? So my research addresses these fundamental yet pressing questions, and more specifically, I ask two related questions. Firstly, why and how does ecology shape the interactions between species, focusing on symbiotic relationship, for example? And secondly, how do the species interactions modify each party to the responses of environmental changes? So today, I would like to start by sharing some of my work and why they are relevant to understanding environmental change. And also in the second half of the talk, I would like to share what the bigger picture is for my future work and how I can contribute to IPCS. So it's my great pleasure to obtain my PhD in the beetle lab with Becky Kellner at the Department of Zoology, University of Cambridge. We study one of the most iconic and fascinating creatures on Earth, the berry beetles, which you probably have heard about it from Dr. Sun's work. But if not, as you can see here, what makes berry beetles so remarkable is their elaborate parental care behavior. In addition, one thing very striking is that they use the dead bodies, such as dead mice or birds, for reproduction. So what actually happened is this. Once a pair of beetles find a carcass, they'll remove all the hair and fold it as a bowl and bury it on the ground. And during this time, they'll be busy laying eggs nearby in the soil. And once the larvae hatch, they'll crawl onto the prepared carcass and then decompose the carcass from the inside out. And so in a crypt made by the parents on the ground, the beetle larvae can feed themselves on the prepared carcass, although they do much better when the parents are around to provide the regurgitation with a mouth-to-mouth contact. So it's really striking, isn't it? So to be able to breed, the berry beetles need to find a carcass about right in size, not too big, not too small. However, life is difficult for berry beetles because carrion is unpredictable and nutritious resource in nature. And all this life means that competition is intense. So there are many conspecifics out there vying for the same food, especially those with larger body size, as well as many other berry beetle species that also compete for the carrion. And in addition to beetles, there are many other competitors as well, such as the blowflies, and the mites. And animals do not live alone, and the mites in particular are associated intimately with the beetles and are commonly believed to cause no harm to the host while taking a ride for a purpose of dispersal. However, they are likely to compete with the beetle when they arrive on the carcass by feeding on the carrion. So both the offspring of the beetles and the mites therefore live together, and once the reproduction completed, the mites of the next generation will climb back to the adult beetles to be transported again. Well, the mites are commonplace in nature in association with many organisms from mammals, birds, or even insects. Whether they are mutualists or parasites remain unknown. And so I address how ecology can tip such a balance between the environmental changes. For this reason, I focus on key ecological factors that can potentially mediate the outcomes of beetle-mite interactions. So then in addition to the interactions within and between species that I mentioned earlier, abiotic environment such as temperature is critical as well, because it largely affects how animals function and behave in nature. And I would like to share with you some of my work focusing on why and how ecology is important in mediating the species interactions in a changing world. And just to give you an idea of what my research is like. And so firstly, I would like to concentrate on the role of interactions within species, namely social interactions. Competition within species are extremely intense because beetles don't like to share and they'll fight off others to guarantee a breeding success. And so I ask, firstly, whether mites do affect the social interactions between the parent beetles, which in turn determine the outcomes of beetle-mite interactions. So in brief, we showed that the mites can team up with the beetles during competition by providing them with the thermal benefits during fighting. However, such benefits are dependent upon the social interactions between the beetles, so that the mites are only beneficial for smaller parent beetles who are doomed to lose the fight against larger rivals, thereby determining the value of beetle-mite interactions. And so let me explain all these in detail. So firstly, I ask if beetles with mites are better fighter. I then spy on them with a video recording as they fight for a carrion in chamber. I then fight two beetles, one with a group of mites, the other without mites, and look at the fighting outcome. And meanwhile, we've also coupled a thermal imaging camera from the top, just so we can record the body temperature as they fight at a distance. So this is how it looks like. Here's a female with mites preparing the carcass on the left, with a thermal video imaging on the right. From time to time, the other female without mites will try to approach, but once the dominant female is found out, she will chase off, and sometimes the fights can be very deadly. We then find that the beetles with mites were hotter, and they're more likely to win a contest. And so how many mites are enough to keep the beetles hot? And I manipulated different mite densities on the beetles and looked at the changes in the body temperature. And surprisingly, the beetles attained 1.5 degrees higher than the background in the body temperature. And furthermore, if we take a closer look on the right, that the smaller beetles tend then to be hotter by carrying the same number of mites. So to find out why this is the case, I've tried to kill beetles and then put mites on, but they did not generate heat, suggesting it is the beetles. And so to be warm, one possibility is that carrying mites makes beetles work harder and therefore physiologically hotter. The alternative is that a group of mites can prevent the heat loss of the beetles as a blanket. So I'm come up with this idea to mimic the weight of the mites by putting a piece of blue text on the beetle and then gave them a little bit exercise. So this is probably the most exciting part of my PhD. So I then modified a tube rotator as a hamster wheel for the beetles to exercise and let them run in a constant speed and measure the body temperature. I can then run every beetle across the control, the weight, or mite treatments here as shown in the black, red, and yellow, followed by a minute and a period of resting. So if mites make beetles work harder, we should see a greater increase when they run with the mites and the weight as well. But in the resting phase, if mites serve as a blanket, we should predict that in the resting phase, that the beetles with mites will stay warmer, but not for the beetles carrying weight or control, because the mites cover a greater area on the beetle. And importantly, we will predict that these effects should be found most pronounced on smaller bearing beetles, because the mites are relatively heavy and also because they cover a greater area on board the beetle. And the results pretty much follow what we predicted. So we show that the mites make beetles warm with a similar effect in the weight treatment. However, mites are also blanket since the beetles with mites stay warmer in the resting phase, but not for the beetles carrying weights. And so by exercising beetles on treadmill, we show that the mites causes beetle to generate extra heat, and then the heat is trapped by the mites as they form an insulating layer on the beetle. So far, we've covered that evidence that the mites do affect the outcomes of beetle competition. So by mediating their temperature, that the beetles were hotter, more competitive, and more likely to be winners. But the question remains if the outcomes of beetle-mite interactions changes according to the social context. I hypothesize that the mites are more beneficial for small bearing beetles. So if you fight small beetles with or without mites against the medium beetle, we should predict that the medium beetles will always win if small beetles have no mites. But if the small beetles have mites, they'll probably have higher or even equal chance to win against the medium beetle. On the other hand, we can fight large beetles with or without mites against the medium beetles. And this time, we should predict to see that the larger beetles will win regardless of having mites or not, because mites are less beneficial for larger bearing beetles. And as predicted, and we show that the mites really makes a difference to the smaller beetles, such that they've got less than 20% chance of winning, but it increases to 50% in the presence of mites. On the other hand, for large beetles, they don't need mites at all because they always win. Having secured the carcass in the first place, however, carrying mites is costly for the large beetles because they then lose the reproduction to the mites. That the mites reduce their reproductive success, possibly because the mites prey on the offspring of the beetles, the larger beetles especially. And now, if we times the likelihood of winning the carcass and the broods they reproduce later on the new y-axis here, we can see beautifully that the mites are in a mutualism for smaller bearing beetles who are doomed to lose the fight against larger rivals. They then degraded as a parasitism to the larger beetles. And so here, just to briefly summarize, indeed, we show that the social interactions can be just as important as many other ecological factors in shaping a species' outcome from mutualism to parasitism. Bearing beetles, however, are not the only competitors on the carrion. They can suffer from natural enemies, for example, blood flies, which are especially competitive at hotter environments. And so in the next study, I would like to move forward to a system where this time looking at how common enemy blood flies can affect how beetles and mites interact in a community context. So over the past decades, we know very little still about the effects of the mites on organisms, especially if they can support the beetles as a protective mutualism. And so this is the first question I would like to ask. And then secondly, as I showed in my previous work, that blood flight competition is mediated by temperatures. And so can temperature also play an important role here too in driving such transition of the mutualism and parasitism? And finally, whether the density of the mites attaching to the beetles can play a role as well in the expression of mutualism in the mites. So the short answer is yes, that the mites make protective mutualism for the beetles, but again, it is context dependent upon the competition from the flies as well as the mite density and the temperatures involved. So just to show you how, we introduce a pair of beetles to breed in the field without mites, 10 mites, or a high density of 20 mites. they were allowed to breed together on a carrion in nature, where the blowflies can come in naturally to compete with them. And at dispersal, we determine the reproductive success for both the beetles, the mites, and also the blowflies by calculating the larval number. And what we found was that when there's no mites, that the beetles and flies compete fiercely with each other. Specifically, the beetles had the highest performance at intermediate temperature, but then suffered at lower and higher temperature ranges, where the blowflies can do quite so well at these temperature extremes. By adding 10 mites to the beetles, we can see that the mites enhance the beetle success, especially at lower temperatures, but for the blowflies that are generally suppressed on the right. Adding a high density of 20 mites, now that the mites not only benefit at lower temperature, but they also benefit the beetles at higher temperatures. But what you can see here is that the beetles suffer from the mites at intermediate temperature where they can independently breed themselves without mites. Again, the blowflies are generally suppressed. As a result, we see that the mites are on a spectrum from parasitism to mutualism, which all depends on a blowfly competition, which is mediated by the temperature, as well as the mite density involved. So the next question to ask is that, how are berry beetles and mites able to cause such a reductions in the blowflies? So the both species wander all over the carrion nest, especially during carcass preparation, before the larvae hatch, and they have been found to consume some of the blowflies when they are still eggs. And so the likelihood that the blowfly eggs will be eaten depends partly on the duration of these vulnerable stages during the development, and also partly on the extent to which the beetles prepare the carcass to compete with the flies. So in the two further experiments, I then tested whether each activity is temperature dependent. And so firstly, we can find that the higher temperatures do accelerate the blowfly larvae development. So the blowfly is spending less time as the eggs and also the first instars throughout the developmental stages than at the lower temperatures. And in the next experiment, we also explore the carcass preparation by the barren beetles. So we set up two cameras, one from the top, the other from the side, and then look at the carcass preparation. We can then extract these photos and estimate the roundness of a prepared carcass by averaging the roundness values. This is because the rounder the carcass prepared means that the beetles are more effective at eliminating the blowflies. And therefore, a value close to 1 indicates a perfect circle made by the parents. And what we really found is that the beetles prepare carcass less well at cooler temperatures compared to the middle or higher temperatures. If you take a closer look on the right, also, that the rounder the carcass prepared, that the fewer blowfly larvae survived in the end. And so the combined effects of the temperature on the blowfly development and the way that the beetle prepared the carcass jointly explain why the blowflies are more of a threat to the beetles at these temperatures. And it is why that the beetles need mites the most. So just to summarize this part, we show that firstly, the mites can indeed be mutualism for the barren beetles, but that the presence of blowflies is necessary to align the otherwise divergent evolutionary interest between the beetles and the mites. And secondly, temperature is important in modulating the transition between parasitism and mutualism. So the barren beetles are forest dwellers that in a perfect world, this is somewhere they live. Until about 4,000 years ago, England was originally covered in the ancient forest. Deforestation began then and eventually created small islands of woodlands. So the natural habitats of barren beetles are not homogeneous. So then in the final work, I ask if beetles are adapted to locally fragmented habitats and what are the underlying mechanisms. So we focus on two neighboring populations in Cambridge, which are just 2 kilometers apart from each other. So there are up to 4 different species of the beetles that can coexist in the UK woodland. and to compete for the same carcasses. In the past 5 years, in Gamlingay, so one of the populations, that the 4 beetle species can consistently be found, and they are likely to compete and arrange their use of carcasses by their body size, so that the largest beetles use the largest carcasses, the smallest beetles use the smallest. Accidentally, during fragmentation, now the oods are separated, and then consequently, Wellesley oods now supports mainly 2 beetle species, so the largest and the smallest, leaving intermediate-sized carrion unoccupied. And so accordingly, I hypothesize that the smallest beetles can now expand their niche to use these intermediate-sized carrion. And so we then take the beetles from the field back to the lab and offer either small or large carcasses for them to breed upon. We then determine how much carcasses are used by the beetles during breeding, calculating as the efficiency as how much carcasses are used for reproduction. And then what we found is that the larger berry beetle species really breed quite well on larger than mice for these 2 larger guys. But for the smaller berry beetle species here, you can see that it's specialized to breed on the smaller carrion. And very interestingly, in the neighboring population, Wellesley, without the intermediate-sized beetles competition, they can now expand to adapted to breed quite well on these larger carcasses. And to find out if such population differences is driven by a process that we called plasticity-led evolution, namely where they can flexibly use different carcass size, we paired up the beetles using a zip-ship design and to breed them either on large or small carcasses. And we can then measure the traits in eggs and also the larvae. And we predict that in the Gamlingay beetles, they may not able to fully use the carcasses because the larger carcasses are normally occupied. And so they lay fewer eggs and generating the fewer offspring. In contrast, in Wellesley beetles, the beetles can expand to use these larger carcasses that are not occupied by the competitors by laying more eggs, generating more offspring. And what we found really was that the Wellesley beetles now consistently lay more eggs than the beetles from Gamlingay, no matter what kind of carcasses they breed on. And consequently, they expand their carrion niche by producing more offspring on the larger carcasses than the Gamlingay beetle. However, on the smaller carrion, they consume the extra eggs that are laid to match the limited resources on smaller carrion they found. So furthermore, with the collaborations with Dr. Shinha and Dr. Miller from Cornell University, we've also detected the adaptive differences between the 2 populations at a small number of genes. And so what you can see here in this scatter plot means that these are genes that are highly elevated in Wellesley populations, and many of these genes are known to be associated with many egg-laying behaviors in most of the insects. Together, I've discovered a remarkable instance of ultra-local adaptations even when the populations are separated only by 2 kilometers apart, with the beetles populations now divergently adapting to breed on a different carcass size due to habitat fragmentation. So coming back to the key questions, so far I've covered how ecology shapes species interactions and how interactions between species can contribute to the responses to the environmental change. And amazingly, I've discovered that the species interactions can be more complex than we previously thought, switching from mutualism to parasitism, depending on the social environment, temperature of the local environment, as well as the intensity of competition. So I've also discovered that isolated natural populations can expand and adapt rapidly to the local environments after habitat fragmentation. And so why do all these matter? And why bother to understand biodiversity in a changing world? This is important because we human beings have fundamentally altered the biodiversity and their functions, on which we all depend to survive. And so what I find most exciting and challenging is the next big question. How do the altered species interactions cascade to influence the community structure and also the ecosystem functions in a changing world? This is my passion and ultimate goal during my faculty position at IPCS. So it becomes increasingly clear and important to understand how abiotic drivers and biodiversity determine ecosystem functions. But we still know very little about how biodiversity is affected by multiple abiotic drivers, thus determining the values of ecosystems. The berry beetles are excellent model organisms for ecology and evolution, but they are crucial too because of their role in the ecosystem functions as they decompose the carcasses in nature. And what we know even less about them is the role of these symbiotic organisms that they interact with. And so as I showed in my previous work, that it might affect the host ecology and evolution, and with good reasons to believe that it might also affect how hosts behave, functions, when they provide ecosystem functions in nature. And so during my faculty position, I also like to extend to a new model system, the dung beetles. So what's very exciting is that the dung beetles too also compete with many beetle species and the rival blowflies. Although the dung seems not good for us, but in fact, the dung beetles consist of a fascinating group and that use the dung as a food resources for reproduction. And they are host for many other symbiotic organisms as well, including many mite species. And so what they do is that the dung beetles fly onto the dung pads and mate and reproduce. And during this time, they can remove all the pests and also help with the waste removal. And as they bury the dung into the earth, they decompose the dung and then thereby help with the nutrient cycling and also support the plant growth and also help with the seed dispersal as they bury the seed down to the earth, just to name a few. And they are also excellent models for biodiversity research in a changing world because they are sensitive to environmental changes and because they are widespread and diverse, that there are more than 80 species that can be found in Taiwan. What's really nice about them is that they can be sampled cost-effectively as important bioindicators, making them perfect model organisms for my purpose. As I mentioned in the very beginning, that climate change and habitat fragmentations are two major drivers for global biodiversity loss, adding another complexity to the story. These environmental stressors do not simply act alone. And in fact, the joint impact of these complex stressors can result in difficult conditions that many animals have never before encountered. And it still remains a major challenge in global change biology. And so to understanding how climate change interacts with habitat fragmentations and their effect on the biodiversity and ecosystem functions, the general framework that I'm proposing is this. So I'll begin with a large-scale field experiments in Taiwan, especially in the central mountainous areas in Qingjin as a representative site, for example. And what you think about Qingjin is this, but actually it's more like this. So I'll target natural habitats with a different extent of habitat fragmentations and to take the advantage of the evolutionary gradients in Taiwan, giving us different combinations of climate and habitats. And I'll then track over time how fragmentation proceeds as they transform into isolated and smaller patches. We can then determine the changes in the insect diversity on the carrion and the dung resources caused either by temperature or habitat fragmentations, and to link that to the multiple ecosystem functions, quantifying as, for example, the carcass or dung removal or the efficiency in the nutrient cycling, etc. Importantly, I will hypothesize that the climate change and habitat fragmentations can interact and predict that they can synergistically impact the insect diversity as well as their functions. So there are several specific hypotheses I would like to test for. And then just, for example, age warming hypothesis will predict that the local microhabitats can easily become unsuitable and unstable for organisms to live, especially at the hotter environments at the forest edges, thereby decomposing, affecting the insect diversity as well as their functions. Alternatively, in a spatial insurance hypothesis, how animals are able to track the changes in the environment through dispersal in nature, but then the habitat fragmentations could impede them from doing so, therefore impacting the biodiversity and ecosystem functions. Crucially, these insects interact closely with a wide variety of organisms, and they rely heavily on the vertebrates as well for the dung and carrion resources. And so my research can also shed light on the impacts of climate change and habitat fragmentations on a network of interacting species through cascades. And you allow me also to investigate how the outcomes of symbiotic organisms change according to different community structures as an underlying mechanism driving changes in their ecosystem functions. Furthermore, my research allowed me to test for a classic hypothesis such as diverse communities can possibly buffer any impact driven by the climate change and habitat fragmentation. And there are two powerful tools that greatly add up the impact and novelty of my research. And so firstly, to unveil the role of the symbiotic organisms, I apply a DNA metabarcoding technique to detect the potential cryptic species in the interaction networks. And so this is because the traditional methods of species identifications requires each specimen to be processed individually. And so it's time-consuming and expensive. But now, the DNA metabarcoding makes it possible to sequence in parallel thousands of DNA barcodes here from a bulk sample containing entire organisms. And so these will also benefit the large-scale and long-term environmental monitoring purpose in my research. And furthermore, to investigate how impacts of environmental changes on the communities cascade to influence multiple ecosystem functions, I applied stable isotope technique that incorporated among the food webs via the animal diets. For example, I'll basically label the carrion and the dung resources. We can then track over time how these labeled molecules flow through the trophic cascades, and to link that to the community structures as well as their ecosystem functions. I'm really excited to bring this new level of research of how organisms respond in a changing world given the complexity of species interactions, as well as their functional roles in ecosystems. But what makes me more excited is that there are many opportunities for collaborations across disciplines based at IPCS. For example, understanding nutrient cycling is critical in my research, but it still requires close collaborations with the earth scientists to work out stable isotope analysis, integrating ecology and environmental science. So then with potential collaboration with Dr. Ren, we will be able to address this challenge and to provide a better understanding of the dynamics and interaction between the biosphere and geosphere, and to help us better predict the responses of the soil ecosystem to environmental changes. Furthermore, I'm also keen to build my local biological processes to a greater spatial scales in Taiwan. Dr. Zhuang's specialty in land use change effect on the climate and environment is particularly timely and can be powerful in predicting how habitat fragmentations and climate change alter the local microhabitats. We can then monitor the responses of insect diversity and their ecosystem functions, targeting the places that are affected most. What has been less emphasized then is to incorporate social dimensions in life sciences, ecology, for example. So my research presents a real-world opportunity to address how biodiversity and conservation efforts should have been made, considering the role of the humans and the society in terms of land use, with a potential collaborations with Dr. Cho, for example. With a remote sensing technique, we can then track back when and how habitats were changed. For example, we can systematically explore which part of the forest to preserve in the modified landscapes, and to protect the biodiversity and the ecosystem functions, and to work out how much protection is needed to be effective to restore these habitats. So together, what I'm really proposing is a major interdisciplinary science that brings together the biological, earth, and also social sciences. Realizing these requires a comprehensive network of the biological responses from my background and links to the ecosystem functions from soil science perspective, and also the impact on the society about how land use and conservation should have been made. All these fit perfectly within the scope of IPCS, since the climate change is never a single issue. And we've got to resolve the practical and scientific problems that can't be addressed purely within each discipline. Meanwhile, I will not limit myself to local scientists, but I would like to broaden my existing research network, including colleagues from Cambridge Zhongxi University, as well as Nanyang Technological University. And very importantly, I've been in touch with the Dr. Eleanor Slade, based at Nanyang Technological University, so we aim to foster the collaborations in research and education in the Southeast Asian region, with this partnership between NTU IPCS and also NTU ASE. So the NTU ASE, Asian School of the Environment, is a world leader in the environmental research focused on the Asian environmental challenges. And very much like IPCS, and I believe that they are initiated both in 2015 or 16 or so, that ASE integrates the earth and environmental life sciences, ecology, engineering, and also social sciences to work out and to address the key issues in the environment and sustainability. And so sharing the same vision, I believe that there are more space that between we two that we can work on to generate more exciting opportunities in the future. To be more specific, I work out plans to establish overseas on-site research opportunity. So the Slade Group has been involved in a SAFE project, one of the largest experimental ecological experiments in the world, based at Borneo, Malaysia. And so the SAFE project, alongside what I'm going to do in Taiwan, can help us to understand and provide a unique opportunity to reveal the complex processes that operate in natural systems, especially in tropical and subtropical regions that contain the enormous values of biodiversity. Having obtained a PhD at Cambridge Zoology, I have rigorous training and experiences in the field of behavioral ecology, which is conceived by my advisor, Professor Nick Davis. And so I'm thrilled and feel privileged to initiate this new lecture, the study of animal behavior in a changing world. I'm also very excited to revive lecturing the climate change biology, which has ceased for a couple of years at NTU. Although there are many similar courses at the moment, but they focus more on the larger scale processes, but I will focus more on the tiny and the detailed mechanisms from individuals, communities, cascades to the ecosystem functions. Ideally, these courses will target the graduate students at IPCS, but I can also customize it for more of a general science for undergrad students to better understand the climate change impact on individuals and cascade to the community and their functions. So what I really love to do in the near future is to engage the general public to gather data with a citizen science approach using the web and the mobile software, for example, iNaturalist. So this means that every one of us can contribute to the biodiversity science only if you have a smartphone. So what you have to do is to record your observations and share it with the naturalist and discuss your findings. Ultimately, by providing insect science to the education with the general public, we'll be able to highlight the importance of climate change more effectively. Altogether, by combining the climate and land use data from the remote sensing technique alongside my detailed understanding of the local biological responses and the ecosystem functions, we can then actually able to monitor and to provide the valuable information for the purpose of biodiversity conservation and also the ecosystem evaluations for the habitats that we don't even know before, and to inform the government and stakeholders in designing effective and sustainable landscape management. Climate change impact on biodiversity is a huge and multifaceted issue, and I would like to address it with the tiny beetles. They're sensitive to environmental changes, and they're widespread in nature and easy to sample, and then very importantly, they interact with a wide variety of organisms that together provide values in the biodiversity and ecosystem functions. After all, it's the little things that run the world. So I hope that I've convinced you how fascinating and important biodiversity is and how it is timely and urgent to protect them in a changing world. I believe that our future depends on the sustainable interactions between the society and nature, and I would like to find out how by IPCS. With these all, I really appreciate all the exciting collaborators from the Cambridge Zoology, as well as the collaborators involved at NTU, National Town University, and also thank these funding bodies to keep my research throughout. So together, my strong background and experiences in the field of behavioral ecology and evolutionary biology will enable me to take on this faculty position with a promising career path. So I'm really excited to join the IPCS because of the breadth and depth that there are many other collaborative opportunities involved. And my research alongside this network of collaborations will help us to bridge the species interactions, communities, and ecosystems under environmental changes, which has never before been more important. And in the near future, what I really hope is that these findings will enhance our ability to predict the impacts on the biodiversity and to identify the species most at risk and to set up suitable conservation and landscape management planning. Thank you very much. And then I got some things to show you. Both some specimens very valuable from the Bering beetles and also the Dung beetles at Taiwan. So please share it and then keep it and I'll have them back in the end. Thank you.
Thank you. Okay, now floor is open. Everyone can ask any questions to the Dr. Sun. Please.
Syuan-Jyun, thanks for your nice talk. So when we talk about the climate changes, so a lot of variables, and particularly atmospheric variables are changing. And in one of your studies shows the effect on temperature. So how about the other variables such as humidity or persist? We know it will change. So how they affect those behaviors as well? Can you give us some introductions, or can you let us know why you first focus on the temperature?
Yeah, thank you.
Sure. It's okay?
Yeah, sure.
Okay. Maybe the follow-up question also, the side conversation we had at the dinner time. A lot of other factors such as not only the humidity but also the air quality, as we had mentioned. So maybe you can express more about the lighting or the air quality or other climate change factors, please.
Yeah, thanks for your question. I believe that the environment is not just the temperature, but there are many factors such as the humidity and other things that you mentioned. But then, in nature, there are many things that are going on. But why I would like to focus on temperature is that it basically determines how fast that insect develop and how they behave and functions in the nature. And so this is why we start with temperature to look at. And then in one of the study, I think the blowflies ones, we also did a lab experiment. So we only manipulate the temperatures in the lab, and we found a very similar pattern to the field, so that we can confirm that the temperature really is a key factor. But I agree with you that, for example, humidity might affect the temperature that an insect experiences. So this is something I would like to explore in future as well. This is why that is relevant to my proposed research on the habitat fragmentations. So imagine that habitat is being deforested, being smaller patches, and so the edge of the forest might become hotter, and so it might become drier, I believe. So that might all impact insect performance and so the functions they can provide. This is something I really love to explore. Yeah, thank you.
And follow that question. So since the temperature is the most dominant one, so it depends on its mean state temperature. So for example, the temperature differences you show, well, it depends on whether it's during the winter or during the summer.
Yeah, this is still a big area that people are looking at different temporal scales in terms of the temperature effect. And I believe that the daily variations in temperatures might affect in the short term insect or animals' behavior. For example, in lunchtime, they might become really hot, and then at the end, at night, it's quite cool. So then they might adjust their behavior and accordingly to the daily fluctuations. But then, given the climate change scenario, it's not just these daily experiences, but the long-term temporal scales. And so it may relate to how fast that insects can generate. So they have a shorter generations, and they might experience this in a couple of generations to these rising temperatures. And so we might expect to see them potentially evolutionary change through these long-term climate change scales. So I think there are two different areas that need to be combined or communicate between the two. Yeah.
Anyone have other questions? Please.
I'm extending Professor Rowe's question. I think it really means that the blanket effect might help the activity of the beetle. However, that's only when the environment is cool. So if I know some animals cannot function well when their body temperature is too high. Okay. So if the environment is warm, like in the summer, then the blanket actually will impede the activity. Is that correct?
So, did you finish?
So, your results only apply to the cool environment.
Yeah.
If the ambient temperature is already quite high, then the blanket effect actually will harm the beetles.
We've started with what the temperature we observed in the natural conditions, and I believe that what you suggest might be true, given the rising temperatures. So if they experience the hotter environments, there's no benefit of having mites and can be even parasitic. I show that larger beetles than the mites and can be even more parasitic for them. So this is something I really love to explore further, looking at the longer temporal scales, whether these rising temperatures might affect how these species interact, especially there's no such benefits around. And then they might just no good for having mites.
Altitude.
Yeah, we can start with that perfectly tolerance.
All right.
Yes.
I'm interested in the relationship between this type of academic finding and actual engineering problem. We probably all know that conserving species is important, but probably it's very difficult to conserve everything, right? So what is the strategy you have to allocate available resources to conservation? How to set the goal, how you use this kind of academic knowledge, and how to prioritize or something like that? Sorry, very vague question, but I'd like to know.
I mean, that's a fantastic question, really. So, yeah, this is still the hot topic, what you have just mentioned. So, of course, there are many species that, at our best, we have to conserve all of them to protect the biodiversity and their functions. But then there are several cases that people identify the keystone species. So, say there are 10 species, but one of them is the key that maintain, say, 80% of the ecosystem functions they provide. And so my research, so linking the individual's responses to the community to these environmental stresses might allow us to detect what species they contribute most to these ecosystem functions, and we get to know them that it is those species who I need to be prioritize our ability to protect them. So I believe that's something can be applied to those area. Yeah.
Okay. Are there any criticism that this way of thinking is too anthropocentric, not ecocentric? Because that function is necessary for humans. Are there such kind of debate in your research field?
Yeah. I'm not quite sure actually, but then when we say the ecosystem functions, it's more like a neutral term, how nature functions in terms of the nutrient cycling and all the functions they provide. But that links to the human perspective that we term it as an ecosystem service. For example, how they provision our life and how they supports the natural system. So I believe there's some a shared interest between the two, and we can start with those aspects to begin with. Yeah.
Thank you, dear.
Anyone other questions?
You think that the result of research applied to urban environment because the very nature of the flowers nowadays so many people live in cities, and we cities try to create a park, and we have died because of a lot of insects inside the city. So do you think this kind of result, research result is tied to
Yeah, thank you very much. I think one of the key topics that I've explored about the habitat fragmentations can link perfectly with what has happened in urban area, because they're less covered, and so they can buffer really quite well in terms of the temperature and other factors as well. And so we can apply the similar approach to look at the urban area, and perhaps we can create the gradients from the urban area up to the kind of hills or the mountainous areas, and to look at the responses of a specific insect and their communities, and if they can function properly in these places. I think one of the good things that on the dung beetle is that they are quite widespread. So you can expect to see there's some species that are vulnerable and they need the perfect natural systems so that they live in the mountainous area. But there are some might be more like a generalist that can be found widely in these urban area and also up to the forest perhaps. And so you can look at what the differences, the functions they provide, and to prioritize these kind of biodiversity conservation.
So how does insect conservation work? Like if you put more of them in than if the climate's warming no matter what, if you're just in the forest and you put more of the key species in, the other parasitic ones are just gonna follow and keep doing their thing, right? So how do you actually make this species conservative? Yeah.
I think, whether conservation is working or not, really needs a long-term scale experiments, and you have to monitor over, I don't know if 2 or 3 years is enough, but then at least the longer scales, and to look at if the populations of these endangered species are really growing or not, and also to evaluate their ecosystem functions, and so that you have the stable populations that perform the right functions in nature. So then I believe that it's not just 1 or 2 experiments can explain all these, but you need a long-term data or even the large-scale global data. Yeah.
It's okay.
If you decide that they need to be helped in some way, what is an example of how you would? Does that make sense? What is an example of how you would deal with a problem physically? Would you just because you can't control the climate of the forest, so what would you do to continue that?
Are you asking about the methodology?
Yeah, methodology.
I mean, how to conduct this research, because according to our IPCS website and we had a conversation with the students, we do the different kind of the methodologies. For example, the model science, laboratory science, and then the field science, for example. So in this research, and how do you conduct research? Because you are doing your PhD in Cambridge, and then regarding to the professionalized question, if you want to move this to the urban forest or the urban ecology, how can you conduct this kind of research into the human-related area? John?
No, I think my question is similar to Sophia, so maybe I can rephrase. I think her question is, if we already know certain type of insects or certain type of species are really important for our ecosystem, and especially in this case insects, and what do we do? And of course we always can do more research. We need multiple years and funding and do research. Even if we already know, say, this dung beetle is super important, so what are some policy or what are some kind of common way people to kind of preserve ecosystem, especially with insects? I think that's your question. What do we do?
Yeah, this is still a growing interest in ecological science, I believe, because what you can basically do is you need the governments, especially, or policy makers, in terms of how much or where these habitats need to be preserved. If I detect from my research these area is particularly vulnerable in terms of the species diversity loss, and you have to set up the suitable decision in terms of how to preserve these, or even create the nearby habitats for these organisms to grow and to form a stable populations. And I believe that's one of the solutions to that. Yeah.
Okay. Okay. Please. Last question.
Question in terms of like your also in the question of conservation. So the selection of keystone species, do you have a certain criteria on how to select them? Or because in terms of like conservation and restoration topics, it's so difficult whether your decision is to prioritize, let's say, insect species that are more resilient in climate change, or will you rather, or some people would rather focus on biodiversity, but yet there are some species that aren't resilient in climate change. So how do you cross between like being resilient and maintaining biodiversity?
I think that is why that lead my research on the species interactions, because they don't live alone. So there's a species you find out what they perform and what their function is, and they are not simply living alone. So they interact with a wide variety of organisms nearby and also cascade to influence the biodiversity in general. So this is why this understanding species interactions can really inform us how they perform or interact at the larger scales and how that altogether influence the biodiversity. So it is not just these single species you need to be preserving, but actually it's more like a closely related species that you need to be preserved altogether. So that's something that, it's just not just the keystone species, but that's a priority. But I believe that you need a better understanding about the interacting species that in the same natural habitats.
Okay. So maybe we save the time. We still have the discussion with the students 1 hour later. So we say thank you again, and for the 5-minute break, and then for the students.